it's going to be nitrogen cycle, which is totally related to what we've just been talking about. It's on page 636 through 637. And then the other one is the carbon cycle. That's 634, 6. No, that's not a 6. 636. And the sulfur cycle, which you guys have already been thinking about because of your Winogrodsky columns, is on page 639 to 640. So not only is this stuff going to fit nice and tidy and on Monday, it's also totally related to stuff you're already thinking about, so it makes it easier to package together with this unit rather than sticking in some other topic. Questions about that? Libby?
It's not so much a question, it's just speaking of lab, that there were some of you who did not streak your plates, and you're still out there on the bench, and you still have time.
For their cell-to-cell interactions? They don't have much time.
No, you don't.
Yeah.
But they've been out there, so please get that done today. I'll pack them up and put them in an envelope jar as soon as I see all of the plates. I think there's 4 or 5 more people who haven't struck their plates, so. Anyway.
People know who they are? You know what we're talking about? Generally, make sure you do it or else your cells won't be growing up on Tuesday. Or is that what we're talking about here?
That's what we're talking
All right, do it. Questions about this? Tony? For this?
Yeah.
Ooh, I could find some. Note to self. Find questions. We're going to talk about this on Monday. It's not like you're just hanging out there on your own, right? I wouldn't do that to you. All right. Okay. Why don't we do our presentations before we get too carried away here? Who wants to go first? Sylvester or Blake? Blake? Where's the slideshow? You know where the slideshow is? You should be able to use either one.
Hey guys, my name is Blake Ziegler. I'm a senior biology major. Today I'm just going to talk to you about a recent article that studied Clostridium hemolyticum. It's a pathogenic bacteria that affects cows and sheep in their liver tissue. And what they found was a really interesting mode of attack and how they invade the cells. They actually use their flagella to attack and cause disease. And so what we know about flagella from this class is that they're basically made of about 20,000 proteins that aid in locomotion and sensing of surfaces. Well, the interesting part of this study was that they found Clostridium hemolyticum to have enzymatic activity in their flagella. So they were able to invade the host, or specifically cow and sheep liver. They were able to break down tissue within the liver of the cow and ultimately cause disease. And so Andrew Doxey and a team of researchers at the University of Waterloo discovered this, and they used bioinformatics and basically found that what these bacteria have done is restructure the proteins in their flagella to include enzymes that can break down tissue. And this has pretty big implications, I would think, for going forward about what we know about pathogenic bacteria and how they can cause disease. Because we also use like bacteria and their enzymes for biotechnology, getting rid of harmful bacteria, or breaking down biofilms. And so my 2 take-home points would be that Clostridium Haemolyticum, and other pathogenic bacteria have enzymes in their flagella that allow them to degrade host cell proteins and cause disease, and that bacterial enzymes are beneficial in biotechnology and can be used to degrade things like biofilms and sticky colonies of harmful bacteria.
Very interesting. So did you read if the flagella are made out of a different protein if the flagella is modified, or do they have enzymes that associate with the flagella?
I'm not too sure. I didn't read into it too.
How would the Clostridium get to the liver? Because your peritoneal cavity is supposed to be
So,
sterile.
So they like discovered the different sequences, and then what the researchers did was actually took the flagella off the bacteria and just put it in an environment with like the cow tissue just to see if they could degrade it,
Yeah.
and it was able to.
Oh, interesting. So it was in vitro.
Yeah.
Any questions for Blake? All right. Thank you.
My name is Sylvester, and today I'll be talking about varicella zoster virus, usually called VZV. So just a description of the virus. It is an enveloped double-stranded linear DNA virus with an icosahedral arranged capsid. It's a member of the herpes virus, and this is how it could look like. So that's a VZV virus over there with an envelope around it. And this thing is known to cause so many issues, but the most popular ones are chickenpox and shingles. So these shingles usually happens after, most of the time it does happen after chickenpox, but it's very, very common. And, so fatigue, body aches, fever, and irritability are the most common symptoms that come before this thing happens. And you usually have rashes that develop on the surface of the body between 200 and 250 inches, like blisters all over. And there's so many myths about this that people who usually get it more than once are likely to die. I mean, if you develop it a second time without any form of immunity, you can die so easily. And the rashes could spread into your mouth, into other intestinal organs. And it usually lasts between a week. Mostly within a week it should be able to vanish if you have strong immunity. But if it lasts more than that, that's when people think it can kill you. And so, if the sickness is very severe, then the rashes could also develop into other internal organs and make it even more dangerous. And the mode of transmission for this is kind of like through air. If people who are infected sneeze or cough around you, you could be infected by it. And also through contact with people who have like open blisters on their skin. And these are images of people who have been infected by chickenpox and shingles. So the image on the left is like a preteen, like around between 9 to 12 years old. And that's how it happens, like you see all those rashes and blisters over there. And the other one is like a 60-year-old person who has been infected by the virus. And those are the most common people that it usually affects, people who are very old, because they have low immunity to so many diseases. So the good thing about it is that it can be prevented through vaccination, and you usually need just 2 doses of the varicella vaccine, and it can cure this. And if you have many rashes on your skin, you can use calamine lotion, which has other carbon in it, and it can help you to reduce it if it's not too severe, but it usually kind of lasts for a while. So my take-home point are that varicella zoster virus is an envelope double-stranded linear DNA virus with an intracellular capsid, and chickenpox is the most common thing that it causes, with shingles as well, and you can use just 2 doses of the varicella vaccine to cure it. People who usually have it do not really get they do not get chickenpox happening, especially if you have. There was a study that I read about, and it said it does not really know if people can actually be cured completely of it. But if you have the vaccine, there's a chance that you can be able to get away with it between 10 to 20 years. So that is the safest way to go about it. That's another thing.
What did you say is in calamine lotion? You said it's calamine lotion containing
It's carbonated.
carbonated?
Yeah, it has it's usually made with carbon
Oh.
and other chemicals in it. At least I've seen one before,
Yeah.
and it's I know a lot of people who have lived around with this kind of situation. And usually when you just kind of scratch it out and apply to it, sometimes it even takes hours, but at most by a day, you see a lot of a quick decline of the rashes.
Is it pink? It's if you use calamine lotion.
Yeah, the color. It's just to reduce the rash, not to cure the.
Did they, someone got it, did they vaccinate against EZV?
They do. So I remember my very first time working at Stanford, I had to go through health assessment,
Yeah.
and I already had MMR, like a vaccination, but it came up that I was susceptible to varicella also, so I needed another booster, which was like an MMRV or something like that. So that was what was done. So they usually recommend that if you don't have very strong immunity, you should get another booster. So that's like the biggest way just to avoid it.
Other questions for Sylvester? All right, thank you. Okay, so I had a question about fermentation. Things to know about fermentation is the point. What do you guys remember about oh, I'm in the wrong mode. I'm going to have to go back and forth today. Themes for fermentation. How would you describe fermentation, first of all?
It doesn't use oxygen.
That was one of the 4 characteristics, right? Fermentation doesn't use oxygen. What else was an important thing? So these are things to know about fermentation. Doesn't use oxygen. Regenerates NAD+. What were you going to say, Ben?
Doesn't use oxidative phosphorylation.
Right, it doesn't have an electron transport chain. Doesn't use oxidative phosphorylation. Those are 3 of the 4 main things. What was the 4th one?
Pyruvate.
Usually uses pyruvate or some pyruvate derivative as the terminal electron acceptor. So that kind of leads to, so depending on where you look, you see lots of different definitions for fermentation. One that you often see is that it is oxidation of an energy-containing compound using an exogenous or an endogenous, using an endogenous electron acceptor. So the endogenous electron acceptor is some organic compound that's part of the pathway that's being broken down. So in the context of fermentation of sugars, the endogenous electron acceptor is pyruvate or some pyruvate derivative, right? So that's all stuff you should know. How about if an organism doesn't ferment sugars? What would be another thing it could ferment? Amino acids. So tell me about that, Christian. What's the deal with that? Glycine and alanine can participate. It wouldn't have to be those 2, but one would do what? One would be the... Should we look at that figure again? That was
23.4.
figure 23.4. Thank you. In this case, they're showing that alanine is getting oxidized right here. But because this is fermentation, that's not really a place where energy is gained. The point of it is that that is part of the reactions here and here that are required to get us down to where the ATP gets made, substrate level phosphorylation. So you don't have to memorize this pathway. But you would need to know that this is a place where substrate level phosphorylation occurs, and in order to get there, you have to have NADH generated. That's really the same thing that happens with glycolysis, right? You have substrate level phosphorylation, the glucose is oxidized, some energy carriers get generated that have to be oxidized somewhere in order for this to continue to get substrate level phosphorylation. You want to look at glycolysis to see, do you have that in your head? Can you compare the two of them in your head without me showing you the picture? I probably did them when I was a junior. Here's glycolysis. Here's a little investment of ATP. That's different for glycolysis. But you down here have the same thing. Oxidation occurs, you get NADH, you have ATP formed by substrate level phosphorylation. Same thing as in the Stickland reaction, which is right here. So in order to continue, and what makes this fermentation is that the NADH has to be reoxidized to NAD+, so that this whole thing can keep going. And in the Stickland reaction with amino acids, it's a second amino acid that serves as the electron acceptor. Glycine gets reduced, it gets deaminated, this comes off of there, and acetate gets produced. So there's a different kind of fermentation, right? All the same stuff holds true. You get this generated, there's no electron transport chain, oxygen's not involved. Glycine and pyruvate really aren't that different from each other. You take this off, they're pretty similar, no big deal. Acetate gets made, which is still a valuable thing, right? So one of the things about fermentation has to do with the efficiency, right? Tony?
Yeah, could you write down what gets reduced, oxidized, and deanimated?
Yeah. So in this is the Stickland reaction. This is the abbreviation for amino acid. One amino acid, these are A's, is oxidized. Another is reduced. The one that gets oxidized is used for production of ATP, by substrate level phosphorylation. That's the abbreviation for. That's not like a universally accepted acronym. I just made that up. Substrate level phosphorylation.
I think the book uses that.
Does it use it too? Awesome. Smart author. Highlight here is this oxidation and reduction uses NADH as an electron carrier. So NADH is made, then oxidized. So reactions can continue. Is that what you had in mind?
Yeah, and you said something about deamination or
Deamination.
Is that important?
Yep. The second amino acid. The one that is oxidized, or the one that's reduced. So in the figure in the book, on page 544, the one that's getting oxidized happens to be alanine. The one that's reduced is glycine. So it wouldn't have to be these particular amino acids, but that's the different kinds of bacteria use different ones. This one is, I think in the book here they're talking about Clostridia, but there's spirochetes that do this. There's all kinds of other organisms that do it using different amino acids. They just have to have a different enzyme to pull it off. So we were talking about efficiency, right? So this is an awesome thing to be doing if that's how you make your energy, unless that's your only choice, because there's lots of energy left in the acetate, right? Because that could go through the Krebs cycle. If you had one, you could go through the Krebs cycle and be oxidized further and make a whole bunch of reducing power, which can make a whole bunch of ATP, but that's not going to happen in this particular case. Questions about fermentation? Yeah?
So for the cyclin reaction, will this occur like whenever it's in like extreme cases of starvation, possibly?
It depends. In some organisms, they just don't use sugar. So it depends on the organism. So it would be most likely, if you had an organism that had all of the choices on Earth on how to grow, the first thing that it would prefer to do is use glucose as a source of electrons and use glycolysis with the Embden-Markov pathway and not the Ender-Deuteroff pathway, right, because you get more energy. Then use the Krebs cycle and do aerobic respiration. But some organisms just don't have the genes encoding the enzymes to do all that stuff. So it's not really, I mean, if an organism had all the capabilities, it would use those first, and then it would use these after all the sugar was gone. But some organisms live in an environment where there's not a lot of sugar, or they're specially adapted to be the decomposers that specialize in using amino acids up. And so some of the bacteria that do this are strict anaerobes that never respire anything, and they maybe can't even use sugar. Tony?
What is the terminal electron acceptor for this?
In this one, the terminal electron acceptor is glycine. Other questions about anything? So I'd rather take time answering your questions than plow through a whole bunch of stuff and then do more on Monday and have you all do crappy on the test. So if you have more questions, it's not like there's any magical amount of stuff we have to do here. All right, growth. We were talking about growth last time. And we were talking about growth in uniform populations. This is what you would call this growth curve. This is what you call a growth curve in a closed system. Growth curve in a closed system. And it's got 4 characteristic phases. Let's look at these since we kind of ran out of time as we were going out. The lag phase, the cells are active and getting ready to grow. The exponential phase, this is where they're growing at their fastest rate. And in a closed system like this, we assume that all of the cells in this growth curve at this point are doing the same things. They're all metabolically active. And then they go into stationary phase, they go into death phase. Interesting thing here is what goes on that triggers this. So there's some things in this chapter that you guys are reading on your own about pH and temperature and environmental conditions that impact microbial growth. Some of that has to do with the transition here. As the cells go from here to here, it's partly because the environment they're growing in has changed, like they've used up all their carbon and they can't grow anymore, or maybe they haven't used up all their carbon but they've used up all their oxygen, and if they're obligate aerobes, they are stuck and they quit growing, or maybe they have made so much acid by fermenting that they've pickled themselves and they can't grow anymore. And that all is happening because they're in a test tube like this, right? Growing in a test tube or a beaker or some closed system, and they don't get anything new and they can't get rid of any of their waste products. So you guys are going to read about some parameters, and I think it was temperature and pH. I want to mention one parameter before we go on because it's going to be related to a couple of other things we're going to talk about. So I'm going to look at my notes to keep myself honest so you know I'm not just making crap up here. The one I wanted to talk about is oxygen concentration and oxygen relationship. So when you think about oxygen relationship, so now we're talking about oxygen not necessarily as a part of macromolecules. This is oxygen in terms of how organisms can experience and tolerate it. And so it's easy for us to forget that oxygen is actually toxic. And the reason it's toxic is because when it gets partly reduced through respiration, there get to be intermediates made that are damaging. And so when we think about oxygen relationship, we think of having enough, but there's some organisms that can't tolerate oxygen, or the toxic intermediates that get generated by respiration. So of course, you guys probably learned someplace that when the Earth was first formed and when organisms started to grow on Earth, there was no oxygen at all, and so everything was an anaerobe because there's no availability of oxygen. And so some organisms have evolved different strategies to handle different levels of oxygen. And so microbes are broken into 5 different categories and described in 5 different relationships with oxygen, which you might have talked about. Did you guys talk about this in the lab on Tuesday? Or did you do that? You didn't get to talking about the 5 different relationships of oxygen, did you?
I did briefly, and then we just tested the facultative anaerobe.
Excellent.
Had a catalase. We're talking about.
Wonderful.
combination,
Perfect.
Something else, but getting rid of hydrogen.
Perfect setup. So we're going to talk about 5 different categories. One is the obligate aerobes. Obligate aerobes. So why would an organism be an obligate aerobe? Metabolically speaking. If obligate aerobe means that it has to have oxygen to grow. Must have oxygen. And the answer to why that would be is so obvious you might not be getting it. It's because they have to use oxygen as their terminal electron acceptor in respiration. That's the only way they can make their energy. O2 as a terminal electron acceptor. And it's obligate, meaning it's obliged to do that. It's its only option. So another category, we're going to talk about why they're like that and what goes along with it in a second here. Second category is facultative anaerobes. So facultative anaerobes are organisms that can use oxygen. They can grow with oxygen, or they can grow without oxygen. So this is a case where if oxygen is available, it may use it and use it up first, because that would be energetically favorable. But once the oxygen is all gone, they have other options for how they can continue to grow and make energy. So they would use oxygen first, then switch to another means. So what other means could they switch to?
Fermentation.
Yep, good. Like fermentation. What, Tony? Yep, or anaerobic respiration. So for example, E. coli, was E. coli the facultative anaerobe you guys used? E. coli would grow and use up all of the oxygen, then when it's gone, it would switch to fermentation. It wouldn't grow as well under those conditions, but it would still continue to grow. Third category are the aerotolerant anaerobes. This one seems a little bit confusing at first, because how could something be an anaerobe and still tolerate air? It seems like that doesn't really go together. The aerotolerant anaerobes are those that don't use oxygen, but can grow in the presence of oxygen. So they're anaerobes because they're not doing aerobic respiration. No aerobic respiration. But they can grow in the presence of oxygen. Okay, we're going to look at some examples of a figure in your book in a second after I talk about the other 2 categories. One is obligate anaerobes. These are ones that must grow in the absence of oxygen. In other words, they die in the presence of oxygen. We'll talk about why that is in a second. obligate anaerobes. And the fifth category are kind of bizarre and a little annoying to me for some reason. They're the microaerophiles. Microaerophiles, they love air, but only in small amounts. They can't grow in a full atmospheric amount of oxygen. What is atmospheric oxygen? It's like 20% or something, Ben?
Yeah, I was just wondering, would the rhizobium be the aerotolerant anaerobes?
Oh, actually, that's a good question. Rhizobia would be facultative, let me see. Would they be aerotolerant anaerobes?
Because oxygen is toxic to them, but they grow in the presence of it?
Oxygen is toxic to the enzyme nitrogenase,
Okay.
but not to the bacteria themselves. So they have to have ways to protect that particular enzyme. But they could grow in the presence of air. I can see why you'd think that. Microaerophiles require a reduced amount of oxygen. Full atmospheric oxygen would be toxic to them, but they respire with oxygen. They use oxygen as a terminal electron acceptor. But if they had the full amount of oxygen in the environment, they would get poisoned. So you guys have inoculated your anaerobic tubes, right? This is a figure in your book that shows the relationship between these. This is in the PowerPoints too. This shows 5 different test tubes, and they have semi-solid agar in them, not totally solid agar. Usually it's a little bit kind of loose. And what's happened is these have been inoculated all the way to the bottom of the tube. So did you guys use the inoculation needles, or did you have them liquidy and then go like this? Liquidy and went like this? So the cells have been dispersed throughout the whole entire tube, and then they only grow in the locations where they're able to grow. So with the obligate aerobes, they only grow right here at the top where there's enough oxygen for them, and there's no growth down here because they don't have the ability to ferment, they don't have the ability to do anaerobic respiration. The facultative anaerobes grow really well at the top until the oxygen is gone, and then they switch over to some less energetically favorable mode like fermentation. And you can tell that from this, the aerotolerant anaerobes, because there's more growth up here at the top. Where there's oxygen, they can grow really well and get a lot of cell mass out of it. Aerotolerant anaerobes grow the same all the way throughout. They don't use oxygen, but they grow if it's there. Strict anaerobes only grow at the bottom. Microaerophiles have a little region at the top where they can't grow, but they grow just below that. So why would it be that why would an organism be an obligate aerobe? Just because it doesn't have the genes to do any of the other stuff, right? It doesn't have the genes encoding the enzymes to reduce nitrate, or it doesn't have the genes required for making enzymes for fermentation. Tony? Yep, they're obligate for molecular oxygen. Otherwise, they wouldn't be growing. They'd be growing clear down here too, right? Yep. Question? No. So what makes them fall into those categories? It has to do with the toxicity of oxygen and whether the organisms have the enzymes needed to completely reduce the oxygen or to eliminate the toxic intermediates. So actually, there's a figure in your book or some stuff in the text here. If I just show this to you without writing it down myself, will you think it's not important? This is important right here. There are 3 different circumstances in which oxygen can become toxic. One of them is when the superoxide radical forms. So in aerobic environments where organisms are using oxygen and they're reducing it with their electron transport chain, sometimes they don't reduce it all the way to what? What would it be if it got all the way reduced to go to water, right? So if they only reduce it part way, they can get formation of superoxide radicals, which are toxic. And in fact, this is one of the species that's used by our white blood cells to kill bacteria that they engulf. They generate superoxide radicals, and it's oxidizing and it ruins the cells, the bacteria. So superoxide radicals right here can get reduced a little bit farther to make protons at hydrogen peroxide, which you know is also oxidizing. It's like bleach. And that's damaging to cells as well. And you guys did the catalase tests, right? So you know that some bacteria have enzymes that can convert this back into less harmful forms. Not all of them do. In fact, sometimes it gets reduced a little bit farther and makes hydroxy radicals. So whether an organism can tolerate the presence of oxygen or not has to do not with the oxygen, but with the partially reduced oxygen intermediates, with the superoxide radicals, the peroxide, the hydroxy radicals. And so bacteria have to have enzymes, if they're going to grow in the presence of those things, to convert them back into a safe form. So an example of that is the enzyme superoxide dismutase.
Could you move your paper?
Yeah, thank you. Superoxide. So that is an enzyme that converts this superoxide radical back to oxygen and peroxide. Superoxide dismutase, SOD. So if a bacterium is making that enzyme and it encounters this, it can revert it back to these things, which are less harmful to it, so it can grow in the presence. Furthermore, it's even better if they have the enzyme you guys talked about in the lab, catalase. Catalase converts hydrogen peroxide back to H2O and water. Is that going to drive you nuts if this isn't a balanced equation? It's going to be a little bit uncomfortable. That would be the balanced equation, right? So the presence of enzymes that do this, there's another one that can do this called peroxidase. That one's easy to remember. Peroxide. Peroxidase converts peroxide to water and NAD+. It uses electrons from NADH. So one of these would be advantageous over the other. Which would be the more advantageous one, probably, if you were a strict anaerobe? Peroxidase would be. Why is that? Because you don't get any oxygen, right? You're not making more of the problem. Tony?
Why is superoxide dismutase turned into hydrogen peroxide? I thought it was toxic.
It is toxic, but oftentimes the organisms that have that would go a step farther with catalase, which brings us back to this figure. So down below these, there's a description of the enzyme content that these organisms would have. And organisms that can grow in full amounts of atmospheric oxygen typically have both of these two enzymes so that they can eliminate all of the toxic intermediates. Same goes for these. They have both SOD and catalase. This one that's kind of in between here, it's not generating, because it's not respiring, it's not generating a whole bunch of partially reduced species, so we can get by with just superoxide dismutase. These guys don't have either one. So they have to hang out here at the bottom where they don't experience any oxygen at all. And this one's kind of in between, low levels of catalase. So from a practical sense, where would you ever find an organism? How could you imagine an environment where an organism would have absolutely no oxygen? Well, if it's buried deep enough in the ground, I suppose it would be excluded, but they're not. They're all over the place. Typically, if an organism lives in a place where oxygen is completely absent, it's because something else has used up all the oxygen. So in the world, there are bacteria all living in a community, right? And the ones that use up the oxygen grow the fastest, and they use up all the O2. They get the most out of it and make the environment anaerobic for the other guys who are stuck doing this stuff, to the point where some of them can't even tolerate being around oxygen anymore. Tony?
These guys have to be like symbionts.
Symbionts in the sense that they're growing with either some other thing or the product of some other thing. They're not necessarily inside of your guts or anything. Right. They're often symbionts in the sense that they're living with other organisms that are modifying the environment in a way that makes it. Well, you could probably imagine that there are places where oxygen is excluded physically, and there would probably be, in places where we would never go, that could happen. Yep, Ben?
Would a good example of a place where you could find anoxic or anaerobic bacteria be in a lake when it becomes anoxic and doesn't have
Yep.
oxygen in it, such
Yep.
as when you get a freeze out in the winter?
Like if you have eutrophication and then you have a freeze out, that's exactly what happens. So the bacteria, the anaerobes in there that, like if you go to a lake and it smells like sulfur and you get, or it's full of methane, methane happens downwards deep too, so that's not just because of eutrophication. Well, it's related, but if you have a situation where there's bacteria in the lake and lots of nutrients, and the lake freezes over, the oxygen gets used up first because that makes sense metabolically, right? And then you have a condition where you have the ability to enrich for the anaerobes that make all kinds of stinking, nasty things that, first of all, kill the fish because there's no oxygen left. It's all been used up. Plus, then they make sulfide because of anaerobic respiration, and they make all kinds of organic acids, and that's exactly right. So, yes. The answer to your question is yes. Well, I'll tell you what. I can see we're not going to make it through the stuff I want to talk about here, so why don't we just be realistic about it and say we're not going to talk about all of the nitrogen cycle on Monday, just so you guys don't knock yourselves out review. How many of you are going to review that all weekend long? I know you're not. Might as well be honest though, right? All right, so questions about oxygen relationship and growth. Yep, Tony? NAD plus. Christian?
For aerosol, anaerobes, and micro aerophiles, would evolution in time start to drive those to be able to consume maximum amounts of oxygen? Like, hey buddy, if you're gonna grow an oxygen, you might as well use it all.
So for the micro aerophiles and the which ones?
3 to 5.
Yeah. Well, actually, if you think of it in an evolutionary perspective, the ability to use oxygen is the new ability, right? You can imagine that you would have, well, here's the thing. All it would take for them to be, in order for the aerotolerant anaerobes to become aerobes, they would have to gain the ability to, they'd have to gain a bunch of genes, right? And bacteria do sometimes gain genes from other bacteria. We're going to talk about that. I mean, I thought we were going to talk about it today, but we'll talk about it next week, right? So they can gain genes. That would be the easiest way, because you wouldn't have to, the amount of changes you'd have to make in this case would be quite a few. This one I'd have an easier time imagining if they just gained a couple of enzymes, or if they change the enzymes they have, they might be able to tolerate oxygen a little bit better. Yep, Tony?
So what's the problem with the micro aerophiles that makes it so they can use such a small quantity?
They don't have quite enough catalase to reduce all of the toxic intermediates. And you can imagine, like Christian just said, if their catalase would catch up and evolve to be a little bit better, or maybe you could just have a change that made it express better. That would be cool, and then you could just have more of it. That'd be easy to do. They're on the spectrum. They're in between the two extremes. Yep. All right. Other questions about that? Okay. We're going to talk about growth in a biofilm. So this is more the realistic mode of growth in the environment. Growing in a pure culture in a test tube or some kind of closed environment like that is pretty artificial. It's more likely that you would have them growing on a surface in the environment. This is the predominant form of growth. So as you learned before, this is growth on a surface. So there's a figure in your book that shows some examples of biofilms. And one of these is a natural place growth of biofilms on the surface of a stromatolite. A stromatolite is basically a fossilized biofilm. It's layer after layer of calcified and fossilized cells that just keep growing and growing. And the surface, you still have living cells that are attached down here. And get their nutrients from whatever floats past them in the environment. Here's an example of a biofilm in a clinical setting. This is growth, a mixture of pus and microbes on somebody's artificial hip. And so there's all kinds of biofilms associated with animals and related to health. They form on your heart valves, they form on your artificial hip joints, they form on your teeth. Dental plaque is a biofilm. It's mostly, dental plaque is mostly cells. Never mind. But if you take plaque from a person, you can tell that it's cells. If there's something in there besides cells, it's very apparent. And so it's mostly the bacteria that live there and their products. And they're really important for all kinds of different industrial applications and cause a lot of problems. And like, we just talked about it. Blake? Biofilm, you were talking about the use of bacterial enzymes to break up biofilms. Next time we're going to talk about the nature of biofilms, what goes into formation of biofilms, and then the interesting thing about it is all of the different metabolic activities that can be going on in biofilms, because you have a mixed culture of different organisms all living in an open system, and so you have a lot of variability in what happens. And we're going to relate this figure to all the stuff we just talked about with metabolism. We got 2 perfectly good minutes. Does anybody have a question for the weekend? You're all on your own and have to learn this on your own. Test is Wednesday. Tony?
You have a question about early reaction.
Yep.
Cyclical reaction.
Yep.
So what's the animation part? Why do they do that?
Why do they do it? So if I were to guess why they do it, I would say the advantage evolutionarily is that when they take the amino off, it actually can be used as an alkylating agent, because these organisms are living in a place where there's acid. And so by having ammonia made, that can be converted to ammonium by taking a proton out of the environment and effectively reducing the acid. It's not used it's not part of the fermentation. There's no importance of it in terms of the actual energy generation. I think it's more of an ecological importance. All right. What? Oh. This?
Okay, would you guys be down for this? I think this would be so fun to have like a potluck breakfast for. Yeah, how fun would that be? Like we can make muffins or something. People could bring eggs. Somebody else could bring
Before when?
like tomorrow morning.
I feel like that'd be your last minute.
Yeah, I know, but like it's not tomorrow. Like,
Typically it is.
Yeah, I think
but
some other
it's
time.
because
Otherwise, you could do that for bonding
it doesn't
sometime.
have low acid. Technically it is, because it's growth surface. But
We could do
depending on
like,
strain,
some of the girls and I were talking and like, we could kind of feel for like something.
they could. Some people use that as biofilm. So as long as they're stuck down, they're thought to be doing something different, but it's probably not the best model. We're going to talk about models on Monday.
And then you
We're
could
talking
do
about other ways.
because then you can actually like,
More realistic.
you know what I mean? Like, it'll instead of like, people, I don't know, it's a little different. That's, we got in trouble because we were like too involved. We what? Yeah. Like me and Billy were in trouble. Because you guys are technically like co-executives. So like if we bring in too many ideas, they get mad at us. We're like, okay. Well, I mean,